A brief about. Dr. Prabhuraj is an eminent agricultural scientist and a distinguished academician specialized in agricultural entomology with an illustrious career spanning over nearly 3 decades. He obtained his PhD in agricultural entomology from UAS Bangalore. Dr. began his professional career as an assistant plant protection specialist at UAS Dharwad, and subsequently he served as assistant professor, associate professor, professor, and head. And presently he is director of research at UAS Raichur. One of his most remarkable contribution in the development of eShop, an electronic pest surveillance and advisory platform, which facilitates the identification of pest diseases and nutritional disorders, and also provides a timely advisory for the crop health. This innovative technology has been adopted across Karnataka, empowering agricultural and horticultural officers and also the benefiting the farming community to a larger extent. Dr. Prabhuraj has been associated with several major externally funded projects as a PI, mobilizing the research support of more than 13 crore from different agencies like Government of India, Government of Karnataka, RKY, DST, and Karnataka Forest Department. His outstanding contributions have been recognized to several prestigious honors, including ICR most prestigious Jawaharlal Nehru National Award, the ICR Best Teacher Award, WSIS 2018 Prize Award instituted by UNO for the ESAP application, and also the Entomological Society of India Best Teacher Award. Dr. Prabhuraj authored an impressive 116 scientific publication with his scholarly work receiving significant recognition from the scientific community. Ladies and gentlemen, with this brief introduction, I would like to call Dr. Prabhuraj a Director of Research, more than that, my UZ teacher. So I would like him to give a lead speak on the harnessing eco-friendly pest management strategies in dairyland agriculture. Welcome, sir.
Good morning to all. Respected the chairman of the present session, the Director of Education, UHS Bagalkot, and the co-chair, my friend, Dr. Venkateshlu, so Director of Extension, and also my student, in fact, Dr. Satyanarayana, so Department Head, and the other, what else? At the outset, so I am very much thankful to the organizers of this very prestigious national conference, so which aptly deals with the dryland and the rainfed horticulture ecosystem, and for giving me an opportunity to share my experience with respect to the pest management strategies which are eco-friendly and economical, so which suits very well to such an important ecosystem. Okay. So with this, let me just start with my presentation. as you all know that, so horticulture is one of the important, the what is called as the ecosystem that India actually is supposed to have due to its very varied agroclimatic the situations for that matter. So any horticulture crop, so be it in a fruit crop or a vegetable or an aromatic or a medicinal plant, if you actually are growing in exclusively in the monsoon situations under the rain-fed condition with no support or a very minimal support from the external irrigation can be referred as the dry land or the rain-fed horticulture system. And if you look at the area under the horticulture system in India, so which closes to the 30 million hectares and producing nearly 377 million tons of the various horticulture crops, which speaks about the richness of our horticulture ecosystem in India we are having. And under this ecosystem, a very particular ecosystem such as the arid and semi-arid regions, which comes under the some of the states of India like the Rajasthan, Karnataka, Madhya Pradesh, Tamil Nadu. So where growing a particular groups of horticulture crops under this special situation will actually provide a very unique opportunity for the farmer to what is called harness the whatever the existing the ecosystem and as well as the rain-fed conditions. Next, please. Some of the important the fruit crops that we normally see it under the arid and semi-arid the regions. The pomegranate, of course, basically was started as a rain-fed, but now is now getting into a very irrigated situation. And bear as a poor man's fruit, what we call it as well as gooseberries, then the custard apple, then the sapota, fig, wood apple, so many. So important fruits which mainly suits to the arid and semi-arid regions. We normally see them growing there. Next. Similarly, under vegetable situations, so we know that very commonly grown the vegetable crop is the drumstick. Though now started earlier started is using as a the pod. Now it has got a lot of commercial value with its leaf for the various medicinal purpose, and also vegetables such as the gourds and as well as melons, which are normally grown under the residual moisture conditions, and some of the beans, the cluster beans and other beans, and also the solanaceous crops are also mainly grown as the rainfed situations. So all these crops, if you look at because of their low requirement of the water and high drought tolerance, okay, and the low input requirement makes it quite suitable for such situations. Next one, please. And of course, we do have the medicinal and as well as the aromatic plants also. So which very hardy and comes under the arid and semi-arid situation. Next one, please. Why they are very suitable for such a dryland horticulture ecosystem, you are already aware, as they are very drought tolerant crop and they actually sustain the prolonged dry period and also high temperature, and provides a very long-term returns. In fact, continuous long-term returns, though with a limited water resources. And diversification is possible in this, where we can grow more than one crop and also provides a sustainability to the farmer, in fact. Okay, next one, please. So important to the farmers from the point of view of diversification, as I told you, that farmer have a lot of options to grow them side by side, and at the same time have an alternative income resources for him. And it also has a multiple usage, not only the fruits which are going to be used, especially in some of the tree crops and the other fruit crops. If you look at, it also provides the various other accessories, including the ecosystem services, so which is quite useful. Okay, next one, please. However, though they are actually drought tolerant and hardy and sustain it in a very dry condition, So the four important challenges that we see them while actually growing. So one, as you all know, are very much aware, and so you are having a feel of it, is the climate change. So a lot of vagaries in the climate conditions is actually putting such these crops in a very hardship, and also to the farmer. So this particular year, as we are aware, with a super El Nino kind of a condition, so the rainfall distribution and the deficit that the India is facing is an what is called as a major example that we can actually give into it. And apart from that, the another major thing is the biotic stress. Biotic stress in the form of pests, diseases, okay, nematodes, so the other non-insect pests, majority of them are there actually. So this session, I will be focusing on the biotic stress, especially the insect pest basically. And of course, other abiotic stress like the soil conditions, okay, the fertility and so other things, and of course, socio-economic constraint. Next one, please. So insects, everybody will agree that they are the major the constraints, and with the different groups of insects which have a different niche areas of their feeding, so it poses a major problem to the farmer, be it any crop for that matter, but especially though in a dryland horticulture system, though we actually earlier used to see a less pressure from these pest complex, but now it is increasing day by day, and also giving a lot of the challenges to the farmer, in fact. So as per the ICR estimate, nearly 6 lakh um, uh, millions of actually the rupees loss has been actually looked at. In some of the crops, we normally see them very 30 to 40%, and an average yield loss to the extent of the 100% basically. Next one, please. I just made a list of some of the important problems or the pest problems in some important crops. Will not go into detail of it. So, for example, if it comes to the chili, majorly we see the sucking pest like thrips, then aphids and mites, so which are major problems in that. And in the brinjal, we get one and only the pest, that is brinjal shoot and fruit borer. And in tomato, we have these fruit borers, pinworms, leaf miners. Okay, so are the major ones, and cucurbits. So fruit flies are the major ones. And in okra again, so we get a sucking pest complex, and as well as the fruit borer complex into it. And coal crops, so we have the diamondback moth and other caterpillars. Pomegranate, as you all aware now, there's a short hole borer, and the anar butterflies and fruit sucking moths. So they have become some major challenges. And of course, other arid fruits like burr and other things, we do get the fruit flies, and some of the sucking pest, and as well as the defoliators. Next one, please. This is just giving you the range of the damage that these problems are creating. But one, in a nutshell, I can say that so they majorly, on an average, we get around 25 to 30%, but it can go up to the 100% in many situations. Next one, please. So some important pest. So I just would like to highlight before actually stressing upon the strategies that actually makes them quite what is called as a vulnerable. So under mango, you can look at the mango hoppers are the major ones. There are different species of mango hoppers, which normally become very severe as soon as the mango comes for the inflorescence stage, and which mainly affects the fruit set. Okay. And then mealybugs, another group of sucking pest, and of course mango fruit fly is one of the quarantine pests that we have. And many a times, and very recently, the Japan also has actually expressed its inability to import mango from India, expressing its the concern over the presence of the fruit fly. And those are the different the damages that we actually look at basically. Next one, please. Then in some important vegetables like brinjal, I told you the shoot and fruit borer, and again the damage ranges anywhere from 10% to the 90%. And I don't go into the details of the nature of damage and all because it's all becomes a very academic actually. Next one, please. Then tomato, as I told you, I got the fruit borer and as well as the pinworm, and of course the white fly, which causes the leaf curl diseases. And so this is another important one. And the fruit borer damage can range up to 20 to 40%, and whereas the tomato pinworm, again introduced a pest and then reaching up to 100%. And whitefly also, if the disease is associated with it, so it becomes quite severe and go to 100% damage. Next one, please. Apart from that, so along with the local or the pest, now we are actually facing a lot of the invasive pest. So chili is the best, very recent example that we have, where the Southeast Asian thrips, that is commonly referred as chili black thrips. So thrips parvis spinus is really causing a havoc in the southern India, especially in Andhra Pradesh, Telangana, and parts of Karnataka, and where the farmers are facing a huge challenge in managing this the chili black thrips. And like that, the papaya mealybug is also another introduced. Then many of them are actually are introduced and become the last 10 years. If you look, so these two are actually stands out and then gives a lot of the importance nowadays. Okay, and next one, please. Okay, so the damage is almost 80% to 90% in any of these cases, if not actually properly managed. Okay, next one. Now, coming to the pest management strategies, here we have to understand one very important situation. The farmers who are actually taking up this crop are actually not as capable, are they? That means the financially. So compared to the other commercial horticulture crops that a farmer takes under irrigated situation. So try to exploit the natural condition and then harvest whatever is coming is the major, what is called the objective of the farmer. So here, so approach should be very, very systematic and it should be from the beginning. So that is what we keep advising the farmer, that you should not or you cannot take the control or the manage the pest only when it comes and then starts causing damage. So we need to be very alert, and they should have a complete knowledge about the problem that they are actually facing, and they should also in a position to expect that when it is going to come and how much damage that it is going to do. So because of that, so it is like prevention is better than cure. So that prevention, the farmer need to start up with the preventing the problem itself. So the first and foremost strategy is to see that these problems are not being actually initiated. So that is the prevention. Then second important thing is that monitoring, survey and surveillance. Every time that they should have a watch and ward in the field and look at the presence or absence of such of these the problems, and then look at and then try to actually tackle it as soon as it is found appearing it. Then third one most important is conservation. See, conservating the natural enemies is the very easy and the most economic way of the pest management, I can say that. Many of farmers will not do that, or they will are ignorant about it. As a result of it, we actually destroy some of the important the weapons, maybe in the form of the natural enemies, which can actually unknowingly take controlling these the pest problems. Okay, so conservation of the natural enemy is the third important thing. And then in spite of that, if you see the initiation or the incidence of the pest, then suppressing with a very eco-friendly and the economic management practices, and go for spraying of a chemical only when it is an absolute necessary. So this is a major strategy that one needs to adopt it, and accordingly they have to manage it. And if they do that, definitely the pest management becomes quite what is called as an cost effective strategy. Okay, next one, please. So how do you actually prevent it? So prevention is, as you know, that is a better than cure, and prevention can be done not exclusively that you need not have to do that preventive measure exactly to prevent or to block the entry of the pest, but along with some of the cultural practice itself, you can actually go for preventing the pests. Okay. So some of the important the preventive measures which actually associates with the cultural practice or so going for the locally adapted varieties which are suitably or suitably pest resistant varieties in fact, okay, or select those varieties which are actually suits not only to the local condition but also they are quite tolerant to the pest. Okay, resistance is a word which we cannot use it or for the pest actually. For the disease this is okay, but for the pest is tolerant is the most suitable one, and they can actually sustain to certain of the extent of the damage by the pest actually. And another important one is that the planting material, whenever we actually get the planting material from outside, we should always see that should be pest free because the inoculum should be avoided from the external source in fact. So some of the classic examples are like the nematodes sometimes which comes from the external source, and they actually sicken the plot or the field and then completely destroy the crop. And going for an optimum the spacing and as well as a balanced nutrition, so making the plant robust, so thereby you can actually make the plant tolerant to the pest. And crop diversification is very important, so growing the multi-cropping system. So we have the horticulture various different multi-storied horticulture cropping systems or different intercropping and as well as mixed cropping system. If you actually grow these crops of a different families which have the different the pest loads or the pest complexes, then usually one crop will act as a barrier or as a deterrent, so for the other crop. In that way the pest load can be actually reduced in that. And of course some of the certain timely operations such as pruning, sanitation, which actually is needed for the crop growth, which also in turn actually help in preventing the pest build up. Next one, please. Next one is the monitoring. The second important thing is that, yes, of course, you have prevented, but you cannot actually keep relaxed that the pest is not going to come. So it will going to come, but a continuous monitoring is needed. Scouting is a very common thing that a farmer needs to do it. And for that, he should have a fair knowledge about the pest habit and habitat and also the feeding nature. So with that, so he can actually monitor through scouting. But the second important thing is that he can go with the some of using the certain traps. Okay, very commonly is a light trap is a common way of looking at the arrival of the pest basically. Or else you can also go with the certain sticky traps. So sticky traps for certain groups of pests, so they are attracted towards certain color. Okay, so that is very important. Say for example, aphids and all are attracted to yellow sticky trap. Black thrips is attracted to the blue sticky traps, more of it. Okay, so like that, if you actually know that, or if you are targeting a particular pest, then you can actually employ that particular the trapping system and then try to monitor and then take a count of it. And another important thing is that food loot traps are there, okay, or the pheromone traps are there. So in case of the fruit borers and all, brinjal shoot and fruit borers do have the very specific species specific pheromone traps, and you can use them for monitoring, and as well as if you just increase their number, then they become a kind of a management or the controlling system through mass trapping. And second important thing is that ETL based decision making. So as soon as you see the pest, so you need not have to hurry to take up the management practice. You always have to know that when it reaches the economic threshold level and initiate the management only when it actually reaches or crosses the ETL. Next one, please. Then conservation of natural enemies. So as an entomologist, I always insist that so you have to actually conserve the natural enemies. You see, natural enemies are those so which are in the various forms such as the predators, parasitoids, or even the entomopathogens for that matter. If you know that what they actually need and when they need it, and if you provide a condition or a shelter for them, then beautifully you can actually maintain an inoculum of these natural enemies in the field. So this will themselves will take care of your pest for that matter. Predators, very common predators that everybody knows, like the lady beetles, so coccinellid beetles, so which normally very effective predators on the soft-bodied insects, such as the thrips, aphids, mealybugs. Okay, so even the small eggs and as well as the caterpillars also they can feed actually, and very easy to identify them and then see that how best that you can actually see they are not be getting harmed. Second thing is the chrysoperla. Chrysoperla is the lace wings, so which again feed on these sucking pests, including some of the smaller caterpillars. And spiders. Spiders are such a generalized predators, and the only thing is you should see that you are not going to disturb them. So disturbance is not a physical disturbance, but through the other means of disturbance, such as the spraying of a broad spectrum insecticide or something like that, are not to kill them basically. Okay, so these are the some of the important predators which actually has a very general predators. They have got a very voracious appetite and they can feed several hundred. Each adult can feed about several hundred the these pest individuals. And in fact, in coccinellids, both adults and the grubs both will feed on this thing. That's an added advantage to that. So as a result, if you simply maintain a few hundreds of these the predators in the field, then they have a good multiplication and as well as a good control of the pest basically. Okay, so that is why you need to look at that and how best you can actually conserve them. Conserving, as I told you, is not to disturb them. That is one important thing. How not to disturb it? So not to disturb it by interfering with their the what is called as a routine activity. Not to kill them by spraying a very broad spectrum insecticides, which are going to wipe out because they are highly sensitive to the pesticide. And second important thing is that you provide a condition for the certain stages of these predators. Say, for example, in case of the lady beetles or the lacewing bugs, in these cases, the adults actually they feed on some nectar and as well as a pollen. Okay, so your main crop might not be actually producing this nectar and pollen all the time. So during the off season, to keep the population within your field, actually, you can grow certain the nectar and the pollen rich the crops, okay, or the some of the plants surrounding the field, and so that it provides a kind of a shelter to those this thing. And this especially holds good when it comes to the parasitoids also. Okay, and avoid excessive fertilizer and irrigation, as well as regular monitoring is required whether that to my field has got a sufficient predators or not. You should keep have a watch and ward over that. Next one, please. Then parasitoids. Parasitoids again, they are actually you will not be able to see them through the naked eye like how you actually observe the predator, but behind the screen that they will be doing a wonderful the job in fact. So parasitoids especially they start with the egg of the pest itself as an egg parasitoid, then larval parasitoid, larval pupal parasitoid or pupal parasitoid, pupal adult parasitoid. You get all stages these parasitoids are going to work out, and very specific groups are there, and it will be very helpful in fact. And this actually parasitoids only for the young ones they need the insect for completion of their younger generation or the larval generation, whereas adults they actually feed on the nectar and the pollen. So as I told you in the case of predators in here also, so providing a shelter crop and an alternative source of the pollen and the nectar will always actually help them to retain the parasitoid population in the field, basically. Okay. Next one, please. Then the third most important thing is the management. Okay. The after conservation. So most of your problems are going to be solved, but even in spite of that, you know that there are very specific pests are there, persistent pests are there, okay, which are bound to occur. Regular pests are there. For that, your approach should always will be the IPM. So integrated management practice. And IPM is everybody knows, and these IPM practices, which is a combination of many possible and available eco-friendly practices that you integrate it right from the beginning. Then with these IPM practices, you can definitely manage the problems. Okay. So those are all the important components of the IPM, and I don't go detail into it. Next one, please. I give I thought of giving some of the case studies for this the pest management, especially the fruit flies, which are common in fruit and as well as in the vegetable. So some of the cultural and mechanical, the physical, the mode of the management, some biological control, all these things are there actually, which is known. But only thing is you know to know that when it has to be intervened. In fact, so for the package sanitation, traps, bagging, bait, and then need-based spray. So for the fruit fly, they have to actually make you stick on to this mantra, such as sanitation, cleaning the field, using the traps, then bagging the fruits, and then baiting the adults through the food lure or the other one, and in that over and over, if needed, some need-based application. Next one, please. Similarly, for the borers and defoliators, so you have some of the important one. Majority I already told you. So you need to actually look at these the management strategies and then do it. In fact, next one, please. Next one. Then the sucking pest, as you already told you that they are the major ones, and we do have the some of the important practices, which is a component of the IPM, and we need to actually use them in a very right method in a right time is a very important thing. Next one, please. Next one. Okay, so this is my last slide. So I just would like to, in a nutshell, I would like to say that so prevention, monitoring, identify, then look at its ETL level, and then act. Okay, so these are the five the things, the points that a farmer should actually look at it. And if at the time of acting, if they found that if the population is less than ETL, then simply has to continue watching it and then look. And if it is found more than ETL, then you can actually go with any of these practices or the combination of these practices one by one. So in a sequential manner, and even in spite of that, if it is actually exceeding, then only we can go with the chemical method. So this is the only way that a farmer can actually effectively prevent the pest complex in the dryland horticulture ecosystem. Thank you. So with this, I conclude my presentation, and if there are any questions, open for discussion.
Thank you, sir, for your valuable and very good present, valuable sharing the information with regard to the diversification and the crops suited for horticulture, as well as the challenges with regard to climate, biotic stress, abiotic stress, and socio-economic constraints. And also, you have covered with the major pests in horticulture crops and the loss due to this pest, and mainly cover the areas of pest management strategies, includes the natural enemies, biological control, and host plant resistance, and also the cultural practices and botanical approaches. Thank you for very sharing valuable information. Now the topic is open for discussion.
Sir, I have one question.
Yes.
Sir, these predators and the parasites, they will attract to some plants for pollen and nectar, like maize, bajra, and sorghum. Is there any commercially available is there, sir?
Is there?
Any commercially available attractants for predators and parasites?
No, there are no commercial attractants as such. Okay.
Yes.
I think for the honeybees, there are some
Yes,
attractants are
sir.
there
Bees,
actually
bee scent
available.
too.
For the
Bee is
bee,
there.
yeah. For these, no, because they are very general in nature.
Yes.
So not specific to any kind of an plant or a kind of an the thing. Only thing is growing certain flower intensive or nectar or the pollen rich plants is more than sufficient. Like coriander, I told you. Okay,
Yes.
some of them are there actually.
If you
But
develop the,
commercially, I doubt there are any attractants for the parasitoids.
if he develops or synthesize some attractants, it is a very helpful, na, sir.
Of
Because
course.
we will manage eco-friendly all the pests.
Of course, of course. Maybe why they are not at come means it may be a challenge for developing a very specific attractant to a specific the parasitoid or as attractant which actually generally attracts the many parasitoid for that matter. Okay. See what happens is you can see in case of honeybees, if the flowering is there to have more bees to get attracted, we are going for a attractant.
Yes.
But whereas in case of parasitoid or predators, if you actually even spray attractant and invite the parasitoids, you should have the source for them to retain them.
Yes.
So without that retention, again the parasitoid might simply get attracted and then again they will go back if they don't find any food there. Okay, so that continuous support will not be there. Whereas unlike in case of honeybees where there is only a specific period of flowering. So in that case, growing the nectar and pollen rich plant itself is the best way of attracting and as well as retaining the natural enemies.
So in
Retention is very important.
yes, sir. In some crop, persistent pests are there now. In that time, we can use, no, sir.
Persistent pest.
Persistent pest is there in some crops. That time we can use the parasites. We'll get host every time.
Okay, okay. So there actually you are telling about the augmentation.
Yes.
Augmentation is like artificially introducing the parasitoids.
Yes.
Okay. So only in such situations, wherever you see, in certain cases of the annual crops, the augmentation or the inundative release of the parasitoid is recommended.
Yes.
But in a more or less perennial ecosystem, like in fruit crops and other things, so there it is always recommended for the inoculative release or the conservation of the natural enemies.
Sure.
That is very important. Like in
Thank you.
vegetables and all, you can have the inundative release. Okay.
Thank
so
you, sir.
that is there.
Any other questions? Yeah.
Sir, I have. So, somewhere we need to revisit the IPM packages available because at ground level, at grassroots, if you closely interact with growers, those who have adopted IPMs in toto, they are complaining in many of the crops they are not working because the concept is very old. I don't know whether it is evolved over a period of time or not. I strongly feel there is a need to assess the effectiveness of these IPM packages across the crops and cropping ecosystems. That is one. And what is the role of community, sir, as a community management in effective implementation of the IPM?
Exactly.
Thank you.
Sir, IPM practice revisiting, if you look at the components of the IPM, okay, and they are going to be remain more or less the same. There is no a newer component into the IPM which can actually is newly introduced into it. We have the same methods or the old of methods of cultural, mechanical, physical, okay, biological kind of a thing. The farmer's challenge here is that two important things. One is that when such components needs to be used, and they are not aware about it or ignorant of it. Second one is accessibility or availability of some of these components, especially the biocontrol components. So because of that, the farmers are not getting confidence in following the IPM practice. But if you look at now the farmer, say for example, here in chili growers, now you now see every field is now actually having these sticky traps. So hundreds of sticky traps they are putting it, and they are quite confident and as well as the faith in that they are going to attract and as well kill because they are seeing the results there. Okay, so it is only that when such components need to be used and also that where such components are available. If they are aware, I think the IPM components are going to be a wonderful work actually. Then coming to the second one is community based. Of course, the community based approach is the best way of approaching any pest for that matter, especially those which are widespread and regular pest. Okay. So like chili black thrips, I was telling you. Okay. And also the tomato fruit borer, something like that, where we make use of certain monitoring and trapping techniques in that kind conditions. So community approach is the best way. So that is one of the important approach. But it has its own challenge in our Indian the agriculture and horticulture scenario.
Okay, so if there are no other questions, thank you, sir, for your nice and informative presentation. So, sir, one small doubt. So we say that most of our the IPM strategies are ETL based, but the application of ETL at the farmer level is as some limitation. So can we think of any other alternative like rational decision making systems in place of ETL, because the ETL has some limitations itself.
Well, sir, actually, ETL becomes a more of an very scientific approach of pest management, which many a times we definitely feel that it is beyond the thinking of this thing. But the same condition as an ETL can be qualitatively converted into a low, medium, and high kind of a situation by providing roughly. So for each of the crop like this, how many fruits needs to be damaged, then only we consider it as a ETL kind of a thing. Number and all becomes a very tricky for the farmer to actually look at it. But visually, if they are able to convert that ETL value into the qualitatively as a low, medium, and high, then that becomes very easy. In fact, for that your matter, the ESAP, the application that we are now actually is now widespread in Karnataka. We have actually brought this low, medium, and high incidence the situation. Earlier, we used to actually deal that in the form of ETL. So looking into the difficulty, we have now gone into that qualitative analysis.
Yeah, okay. So thank you, sir. So any other questions? So if there are no questions, so I thank Dr. Prabhas, sir, the Senior Professor of Entomology and the Director of Research of US Raichur, for his valuable presentation and the very good discussion. Thank you, sir. Thank you very much.
Thank you very much.
I request Dr. Pakruddin sir, Director of Research, UHS Bagalkot, to felicitate Dr. Prabharaj sir. Thank you, sir. The next speaker is Dr. Raju B. Teggihalli. I would like to give a brief introduction. Teggihalli is presently serving as a senior scientist and head and program coordinator at ICR KVK Kalaburgi under University of Agricultural Science Raichur. His professional contributions reflects strong commitment to agricultural science from laboratory to farmer's field. As an entomologist, his research and extension activities have focused extensively on crop protection, integrated pest management, sustainable agriculture, and eco-friendly pest management strategies. Dr. Teggihalli has made significant contribution towards management of important insect pests affecting the crop, particularly in pulse-based cropping system. A major strength of his professional career has been his dedication to agriculture extension and to transfer of the technology from laboratory to the field through KVK system. Dr. Tegirli is also actively involved in guiding the youth farmers, especially in Kalaburgi areas. With these brief background, I'd like to call upon Dr. Teggihalli for his lead talk. Welcome, sir.
Respected chairman, co-chairman, and my dear and DN, all officers of this university and our university, and my colleagues. Today I'm going to present the things what I have learned, our KVK has learned from the climate resilient technologies which we have demonstrated in the farmers field. The technologies which created an impact on that day. I'm presenting some of the slides regarding this technologies. Next. So, Rahul, next. Sir, as you know that our Kalburgi is totally a dryland area where it is called as also the pulse bowl of the Karnataka. Majority of the pulses are grown there. And apart from that, only the horticulture component what we have is only 1.9 to 2% of the area is occupied by the horticulture crops. That is very notable thing. And apart from that, we have the small, the soil characters, less than 0.5 organic carbon and erratic rainfall. These are some of the situations like that. And among the horticulture crops also, banana occupies a major area apart from the dryland horticulture. Next. So this was the impact. Actually, we were earlier working with the farmers. This is a red gram crop. Actually, one technology what we demonstrated is demonstrating on the sowing of pigeon pea on ridges and furrows. Ridges and furrows means the furrows, as we all know that the furrows act as a conservative furrows and excess moisture will be drained out. And without the normal sowing, we can see the crop. This is the impact what it has created. Actually, the greenery crop, what we are seeing, it is the crop which is sown in the ridges and furrows. And the other side, which is a dried one, it is a totally a dried one due to the scarcity of the rainfall. Actually, pigeon pea requires about some little amount of the rainfall during the October month. If we sow on the ridges and furrows, this will be very good impact. And based on this, and again, we started thinking on this not only on the red gram, we should think on other crops like dryland horticulture crops. So in the next lesson, what we have learned is next. The next, sir, this KVK, we contacted a lot of the donors and all these persons to execute our programs. This KVK has listed out all the resilient technologies which are suitable for the dryland horticulture crops. And this for this horticulture crops and the funding is very, very important. That's why we contacted the funding agencies and the list of the technologies we have demonstrated in the farmers field. So that thinking that this dryland horticulture will permanently help the farmers of this village. Next one. So what we did is locally suitable dryland tolerant fruit crops under the rainfed like jamun, mango, guava, and lime. These crops we have taken. And also we are focused on this, not only the concentrating on the crops, we focused on the moisture conservation to retain the crops for the next years. So the intervention was the long-term climate resilient technologies, not only for planting and this. That's why we adopted some of the technologies for the long-term asset creating like that. The next one. We can see here, the first one is the dryland horticulture crops, the jamun, lime, guava, mango. And the amount generated was about the 18 lakhs. It was funded by the other agencies. This Ashraya trust agencies they have given. And number of farmer benefited was about the 88 farmers. The village is called as Suntnur village, and in adjacent villages, we took this plantation methods. And after that, we trained the farmers how to conserve the moisture by applying the mulch like this and using the drip. And again, also we faced the several problems. Next, the challenges were that. The next one, apart from this mulching, watering, and all these things, and we faced the problem of the water. Water was a major constraint here. The next thing what we did is we again contacted this next one, sir. And we made the tanker one. We use the tanker to irrigate all the water and like that. And again, the farmers placed that they should have the drum and all things. That's why we made a permanent structure. This is a drum which is a capacity of cement rings of 1,000 liters capacity. And we used to bring the water from the other places and dump in this every farmer's field like this. And the farmers used to water their plants by using this small pipes or by rose can like that. This was the thing what we have done. Again, we faced the problems because bringing the water from other side and dumping in the 2 months, it was very, very difficult. Then again, we thought of another thing. Next one. These were about the 50 cement storage tanks for the 80 acres. The tractor, we used to give the tractor and use per 100,000 liters. This was sufficient. But next, what we did, they felt the difficulty like this, sir. Even though they have the dryland crops, they faced the difficulty of the watering, and the water was again the scarcity. bringing the water from other place was a difficulty. Then again, we thought of the another, why can't we regenerate or recharge our wells, bore wells and like that. And the next again, we went for the recharging or the recharging or desilting. And that in between these crops, sir, this is one of the interesting story. In between these horticulture crops, earlier we used to sow this sorghum. Sorghum, Madi, it was a very drought year like that. Rainfall was, we didn't receive any rainfall. And that time we watered the sorghum day after the flowering. Then he got the completely yield, very good yield, about 3,600 per acre approximately he got. Apart from this, apart from the fodder and all that. This is a very good technology, what we demonstrated in between the horticulture, dryland horticulture crops. The next. And again, this was some of the photographs what I wanted to show. The next. And in the 60 acres, what we promoted was about the convergence was about the 18.2 lakhs amount was generated when the 4 major crops, sir, and the technology provider was from our UH, our university, and the KVK implemented this one. The convergence was made with the Ashraya Trust and also the Marada NGO, which helped the KVK to select the farmers and to train the farmers and to mobilize the farmers and like that. By convergence and all those things, we did like this. And the next one. And then we thought again the water, it is problematic. So we started of thinking of desilting the open well, desilting of open well, and the community farm ponds we have to do. And the check dams, we have to again desilt the check dams and the compartment bunding for sorghum rabi crops like that. These are the technologies what we have demonstrated here. So we started the desilting the existing wells. Actually, that village is a Suntnur village where about 100 wells were there, but all wells were dried up due to the silting. Then we thought, why should not we do the desilting? And then we started for making the desilting of the wells. The next. You can see here, sir, the machine was actually from about the farmers were about almost the contributions from the farmers was some of the amount was contributed by the farmers, and the rest of the amount was given by the Marada NGO, and from KVK also very small seed kits and like that we were giving. The totally was about the 25 lakhs we have spent. But when we see the capacity of the water, sir, about 65.54 lakhs liter was harvested, was again regained from the well, and it was sufficient for to irrigate about 131 acres of the land. This was the thing what we have done. We have desilted from 100 wells like that. Next. We can see here before and after. Before it was a very small thing. After we get the water, about 100 wells we have desilted. The next. And this was a check dam, sir. Just I want to show you these figures. The earlier situation was like that. And the width of the check dam was about almost 12 meter and length was 55 meter and 1 meter depth. You can see the capacity of the water. It will hold about almost 6 lakhs, 60,000 liters of the water it will hold, sir. Then by using this nala, then they have started the growing banana as well as onion. They have started growing again the critical irrigation for the pigeon pea and sorghum. Such of the experiences we got from the desilting of these nalas. The next one. And these are some of the pictures what you have sent before and after. Before we can see, sir, and after it was very clear that, and we are able to provide the water to many of the farmers. Next. And these are some of the photographs indicating the before and after the check dam NRM activities, what we have taken at KVK. Next. And these are the sums. So totally about the 9 lakh liters of the water were harvested. There we have harvested about the 31 acres from the irrigated farm wells. And from these nalas, the capacity was harvested about the 9 lakh liters. Almost the area where the irrigation can be provided is about 20% of the field of that village can be provided with the irrigation. The farmers started growing the pigeon pea again, leaving the pigeon water, irrigating the pigeon pea for irrigating for the pigeon pea. Again, they get the good yield. And again, they have started the growing of again other horticulture crops. Next one. And these are some of the pictures, the check dam constructed and total area irrigated. Of this is some of the data. So the next one. And the before and after, I included some of the photos, check dams which were constructed, and totally overall it was about the 68 acres of the land which was brought under the dryland cultivation in the Suntnur village. And this is apart from the irrigating the agriculture crops like pigeon pea and sorghum. Next. And this is the impact of desilting of the borewells and the results of this one, sir. and the desilting of the check dam, which happened to be about the 60 acre of the pigeon pea and the 20 acres of the onion. Because in the pigeon pea, actually what happens during the month of this October or starting of November, if you do not receive the little quantity of rainfall also, the pigeon pea, it will go for the wilt. Wilt will be affected. That microfamina wilt will be affected. Last year, about 300 crores of the compensation has been given by the government. Looking to that, we started demonstrating that at the time of the critical irrigation system, we will irrigate and got this good result. And opening, desilting of open wells, this is about the 220 acres pigeon pea and 100 acres of onion, apart from the horticulture crops. Next. And some of the people which have started growing the banana and onion, and again the irrigation for the wheat and total vegetables on 3 acres. One of the farmers' experiences like that. Next. And this for this compartment bending for the sorghum, we have also demonstrated for this sorghum compartment bending. This is also one technology which is giving up the very good results. Right. Okay. And these are the results of this compartment bending, what we have when it was received. And actually it is about the 10.5% of the increase in the yield was there. Apart from we getting the good quality fodders. Next one. Yes, Mohit. The results, and this was the results actually about the 60 acres of the dryland horticulture we fought. Conservation, this convergence support was about the 18 lakhs, and overall storage was about 65.54 lakh liters of the water. Estimated crop benefit was about almost 1 crore, like 97 lakhs. And this is due to these are some of the photographs. After 3 years, we are getting these results like this. This is the recent photos. And 60 acres, we will find the dryland horticulture crops, which is a intervention of KVK. And the climate resilience, what we have the project has created is, it is reduced the dependence of the single crop, and the moisture conservation for the better use of the conserved moisture and the groundwater, especially in agriculture crops, mulching and ridges and furrows, that technology was demonstrated. Apart from that, risk reduction to improved capacity to withstand the rainfall variability of the horticulture crops. Asset creation, this is a very, very important thing. What we have done, about 60 acres of the horticulture orchard has been established. The horticulture becomes the resilient when it is embedded with complete water, soil, crop management. This is very, very important. Not only on the dry planting is not sufficient, it have to maintain this soil and crop management aspect should also be taken care. These are the lessons what we have learned. Select the fruit crops which are suitable for the soil, rainfall, water availability. Combination of this horticulture and water harvesting structure for moisture conservation. This is very, very important thing. And conservation with the essential scaling and resource. Now we have submitted one project for KKRDB by looking to this pilot project Bandhu. Demonstrations and repeated visits of this will improve the farmers' capacity very much. This is very, very important thing. Demonstration and repeated field visits, sir, for improving the knowledge of the farmers. This is very important. And long-term handholding is the critical during the orchard establishment. And these are the document outcomes and outputs. This is the thing what we have done, NICRA experience and the dryland horticulture, what we have introduced, water conservation and conservation. These are the key points what we have worked on. Thus, and again, we will look for the other villages to work on such projects. So this is what I wanted to tell. Thank you very much for providing the opportunities.
Thank you, Doctor. Have nice presentation with regard to approaches as well as the dryland horticulture intervention, especially for the implementation of the long-term technologies. And also you have covered model and implementation of the program, as well as the water and soil conservations and convergence activities. Thank you for your nice presentation and informative.
Thank you, sir.
Now the topic is open for discussion.
Okay, if there are no questions from the audience, so thank you, Dr. Raju Tegeli, for
Thank you.
your nice practical the presentations where the climate resilient activities have been initiated. So since how many years it is running?
For 5 years, sir, 2021.
Yeah, it is a very nice the impact you have created. And so are there any, because there is a crop diversification and action is also increased.
Yes, sir.
Are there any backward and forward linkages to be created in your
Sir,
project?
it has to be created, sir, marketing of the all fruits,
Yeah.
and we have created on FPO also, sir, for
Hmm.
regarding the horticulture, but we have to go forward. Not right now, it is not
So
existing.
the
We are
production is
thinking.
not
No.
because we're not coming. So
No.
maybe in the days to
In
come.
future, we are thinking
Yeah,
of that
yes.
marketing.
Thank you, sir.
Thank
Thank you.
you.
Very nice
Thank you, sir.
presentation. Thank you very much.
Thank you, sir. Now I request chairman of the session to felicitate the speaker. Girish. Thank you, sir. Now I hand over mic to chairman of the session to conduct the oral sessions of this particular session.
Thank you, Manjunath. Now I request the Ramesh to present the topic, that is eco-friendly management of twisted disease of onion using bacteria. The time allotted is 7 minutes, so you have to cope up with the timings. Yes, sir.
Good morning, respected chairman, co-chairman, and all the delegates. So myself, Dr. Ramesh, assistant professor. I am here with presenting the presentation on eco-friendly management of twisted disease of onion using endophytic bacteria under the theme 5, that is ecological pest and disease management horticultural crops. So this is about the introduction about the crop and the disease. That is onion. It is belong to family of Amarlidaceae and it is called as queen of kitchen. And also in Karnataka, in India, the Karnataka is the third position in the onion production with the area of 51.9 thousand hectare. And major onion growing districts in Karnataka, Vijayapur, Bagalkot, Gadag, Chitradurga, Chikmagaluru. And also the major constraint in the rainy season, the twisted disease. Because of this disease, the 70 to 80% of yield losses reported. And the management of this disease, only we are depending upon the chemicals, the fungicides that may lead to cost effective and also the risk of residue problem. With this background, we selected the objectives that isolation and identification of bacterial endophytes from the onion, evaluation of isolated endophytes against a twisted disease pathogen under in vitro and field condition, and also to study the secondary metabolites produced by the effective endophytes. So these are the materials method. The first is survey conducted and collected the healthy onion sample from the twisted disease infected field. And second is isolation of the bacteria using the tissue plating method. Third is cultural and morphological characteristics documented these isolated endophytes. And fourth one is in vitro screening of these endophytes against a twisted disease pathogen using dual culture method. And fifth is field evaluation of the effective isolate. So sixth is that secondary metabolites profiling of the promising isolates. So this is the symptom of the twisted disease. Whether here you can see the white color spots, the spot is enlarges, and you can see the salmon color, the fungal body development. The number of spots increases on the leaf, neck, and also on the bulb, that may resulting in the twisting of the leaf, neck, and bulb. Apart from that, this symptom, so without seeing any spot, the plant will also be twist and enlarges the leaf and neck region, that may leads to no formation of the bulb and rotting of the bulb. So here we isolated the pathogen Coltotrichum gliosporidis, Coltotrichum truncatum, Fusarium oxysporum, Fusarium fusicari, and Aglothecia ralphs. From this, I can say that this is a complex in nature. And the survey conducted totally in 7 districts. From there, we collected 46 samples. From there, we isolated the 46 bacterial endophyte isolate using nutrient agar media. Afterwards, that the cultural characteristics like colony color, colony nature, margin, opacity, texture, shape, and the gram reaction. These things we documented. Then further, the in vitro screening of endophytes against this twisted disease pathogen. So here the dual culture method we used, and among the 46 isolates, the isolate number E17 and E37 was showed maximum inhibition against the Coltotrichum gliosporidis, and also in case of Fusarium oxysporum, the isolate number E17 and E37 was shown the maximum inhibition, and in case of Aglothecia ralphs, also the isolate number E17 and E31 was shown the maximum inhibition. For this studies, we only selected the isolate number E70 for further study. So this is the pure culture of E70. And here the biochemical characterization of the E70 isolates. Here the gram staining, stars hydrolysis, KOS test, oxidase test, and indole test. From this biochemical test, it was confirmed that it is a gram positive in nature. And further, you can see there are some imaging of this bacteria. It is a rod-shaped bacteria, and further, the molecularly it was confirmed by using the gene that is 16S RNA, and also housekeeping genes, that is a gyrase A, gyrase B, and the reg gene. It was confirmed that it is a Bacillus velegenesis, and the gene was sequenced, and the sequence gene was deposited in the NCBI gene bank, and we got the accession number also. From that accession number, we drawn the phylogenetic tree. And finally, the field evaluation of this effective isolates, that is Bacillus velegenesis. Here, the totally the 7 treatment. The one is the 8 treatments with the 3 spray from the onset of the disease to the 10 days interval. And the T1 here, propiconazole, T2 here, the combi fungicide, azoxystrobin, tebuconazole, T3 is the Bacillus velegenesis, T4 here, the first spray of Bacillus velegenesis followed by 2 spray of propiconazole, T5 propiconazole followed by the 2 spray of the combi fungicide, T6 Bacillus velegenesis spray followed by combi fungicide followed by propiconazole, T7 propiconazole, then combi fungicide, lastly velegenesis. So the 2-year experiment we conducted at the Haveli. So that from that the PDI after the last spray here recorded the T2 treatment recorded the lowest PDI in both the year, which was followed by on par with the T6 and T7, means that any among the 3 spray, the one of the spray is this bioagent. And also the next best treatment here is a T3, that is the spraying of the endophyte Bacillus velegenesis, 3 spray at the 10 days interval. Apart from that, the in case of the yield, so here also the T2 was recorded highest yield, followed by T6 and T7, and the next treatment is best treatment is T3. So then secondary metabolite profiling of promising isolates by using a LCMS method. Here the totally the 78 compounds we got. From the 78 compounds, the 8 compound having the antifungal properties, and these 8 compound, so we purchased the based on the availability in the India, the 4 compound, that is furphenol, undecanol, butylated hydroxytoluene, and phenol. So the here we and also all these 4 compounds, they are dissolved in the acetone. So that's why we kept as acetone also one of the control, and also in vitro condition using a poison food technique. And here the among the screened against all these pathogens, so the butylated hydroxytoluene and phenol at the 100 ppm concentration, it was so the 100% inhibition. Then again, we reduce that concentration to 50% and 25%. There also the butylated hydroxide was show the best inhibition against choledotrichum and sclerotia. And also at 25 concentration also, it was shown the better result. From this, I conclude my presentation saying that the twisted disease of onion, it is a complex in nature. That is Bacillus velegenesis by spraying alone or a combination rotational spray with fungicides and fungicides. The policy implementation or recommendation here, I can say this Bacillus velegenesis, the gene was sequenced and it was deposited in the NCBI bank in the university name. And also the first year farm trial on this Bacillus velegenesis was completed at the Vijaypur, Koppal, Bagalkot, and Kumbhapur. And the second year result, we are waiting. After getting the second year farm trial result, it may be recommend any of the treatment for the farmers level recommendation. Apart from that, the further study on the butylated hydroxytoluene and the phenol for the formulation development and dosage standardization and also environmental safety issue. So thank you.
Thank you for nice presentations. So you have covered with the complex disease of onion, and also this disease, it ranges from 70 to 80% because of one of the important and major disease in North Karnataka. So you have covered most of the etiology and management aspect, and also the confirm the biochemical test for identification of the effective endophyte and also the antifungal compounds. Okay, very good. Thank you. Now the topic is open for discussion.
Any questions from delegates?
The disease
Any data, because it's one of the severe disease last year, because you have heard that along with you surveyed a lot of area.
Yes, sir.
If you have any data of taluk wise or district wise percentage with respect to percentage.
Yes, sir, it is there, sir. Because of the time shortage, I am not presented here, sir.
Yeah.
The 2 years data, survey data.
Okay, you have the data. Okay,
Yes.
good. Ramesh, I have one clarification.
Yes,
So
sir.
most of your treatments are like a curative.
Yes, sir.
So why you have not adapted any prophylactic the treatments?
Yes, sir. That is the seed treatment and also the treatment for soil application. So that those things we given for the next students.
You have isolated the bacterial native isolate you have taken.
Yes, sir.
So instead of blending it with the fungicide, so you could have tried like multiple times spraying the same thing.
Sir, that treatment also it is there, sir. T3 treatment.
How many times did you spray?
3 sprays, sir.
With
From the onset of the disease to 10 days interval.
10 days.
That also shown the next best treatment, sir.
Next
It is
best.
not on par with the fungicide, sir.
Fungicide not with the fungicide.
Not on par, sir. But it is best treatment, next best.
But you could have increased the number of doses also.
Yes, sir. That
Frequency also, like so that it can come in close close proximity with that of the fungicides, because like fungicides there are so many.
Yes, sir.
And cost is the one thing. Apart from that, like control after a few days, like it is developing resistance. This is what exactly we are seeing in the field.
Yes, sir.
So instead of 3 sprays, you are taken up, isn't it?
Yes, sir.
So that way you could have taken up the 4, 5 or something like that.
Yes, sir. Apart from that spray, number of spray increase, the seed treatment and application of if I am directly soil application, that also be includes, sir, in future research program.
Okay, thank you.
Thank you, Ramesh.
Thank you, sir.
Now I would like to invite Shruti P. Development of endophyte formulation for management of mango anthracnose.
Good morning, everyone. I am Shruti Paul, a senior MSc student from the Department of Plant Pathology. I am here to present on exploring of endophyte for management of mango anthracnose. As coming to the introduction part, a mango scientifically known as Mangifera indica from the family Anacardiaceae. It is known as the king of fruit, as we all know that because of its highly nutritious compound and antioxidant rich in mango. And among all the disease, anthracnose is a major fungal causing disease. It is caused by the Caletotrichum gliosporidis. To manage the disease, we adopt many biological control methods, and among that, endophytic bacteria and fungi will offer a promising potential for management of disease. So my study focuses on objectives like isolation, screening, and characterization and identification of endophytic fungi and bacteria, showing the antagonistic activity against Caletotrichum gliosporidis. Coming to the materials and methods, we isolated the sample. For endophytes, we chosen a healthy mango leaves, petiole stem. But for pathogen isolation, we chosen the sample which is infected with the anthracnose symptoms. Later, we done the surface sterilization with the standard isolation procedure. And we have done isolated the pathogen, that is Caletotrichum gliosporidis. And later we have isolated the endophytic fungi and bacteria. And we checked the antagonistic activity through dual culture plate assay. And later we done the characterization, cultural, morphological, and also biochemical. And at last we done the molecular characterization. We use the 16S rRNA region for bacteria and ITS region for the fungi. Coming to the result part, first we isolated the pathogen from the diseased part, and we inoculated in very healthy seedlings with no symptoms at all. And then we keep for incubation for 2 to 4 weeks. After symptom development, we re-isolated the culture, and we can observe the conidia and the culture plate of Caletotrichum gliosporidis. And these are the list of fungal endophytes isolated from the 12 different varieties from the different plant parts, such as leaf, stem, petiole, and flower. So total, there were 35 fungal endophytes. And we also isolated the bacterial endophytes. There were total 43 bacterial endophytes from 12 different varieties from different plant parts, such as leaf, flowers, petiole, and stem. And also we checked the antagonistic activity for all the 35 endophytic fungi. Among all 35 endophytic fungi, UHSCF9, that is from Khadar petiole, was showing more inhibition percentage, that was 76.11%, and it was followed by UHSCF4 and UHSF10. We can see here in pictorial image all the 35 fungal endophytes are checked for antagonistic activity against the Calitrichum, and it is compared with the control plate, it consisting only pathogen. And also we checked the antagonistic activity for all the 43 endophytic bacteria against the Calitrichum. Among all 43 bacterial endophytes, the UHSB11 and the 14, that's from Alfonso petiole and Khadar flower respectively, shows the same inhibition percentage, that is 94.44, with the less mycelial growth 5 mm. So in pictorial image, we can see that all the 43 bacteria showing antagonistic activity against the Calitrichum compared with the control. And also we later done the characterization with cultural, morphological, and also the 17 biochemical tests were done for the effective endophytes just for UHSB11 and UHSB14. And also we done the molecular identification. PCR amplification was done for pathogen, that is Calitrichum gliosporidis CG1, and for 3 effective endophytic fungi, that is Pseudofusicoccum veliosum, UHSCF4, and Pseudofusicoccum adenosoni, UHSF9 and UHSF10. And also PCR amplification was done for effective bacterial endophytes, that is Bacillus pumilus UHSB11 and Bacillus altitudinus UHSB14. So the after amplification, the bandwidth was approximately 600 base pairs. And we also constructed the phylogenetic tree for the pathogen, that is Calitrichum gliosporidis CG1. We can observe that CG1 is closely related to the Calitrichum gliosporidis strain JEX19. And also we constructed phylogenetic tree for the effective bacterial endophytes, that is for UHSB11 and UHSB14, which was closely associated with the IRH, that Bacillus altitudinus strain IRHB180, and for 11 it was closely related with the Bacillus pumilus strain UM10. And also we constructed phylogenetic for the 3 effective fungi. For EHS and 9 and 10 were closely clustering with same, but UHSF4 was clustered with the Pseudofusicoccum valians strain GHF1. With this, I will conclude by saying that I isolated total 43 bacterial and 35 fungal endophytes respectively. For bacterial endoconistic activity, that was ranged for 57.5 to 94.44 inhibition percentage, but for the fungal, it was ranged from 13.05 to 76.11 percentage of inhibition. So later we done the molecular characterization. So we identified that effective isolates were belonging to the Bacillus genus and Pseudofusicoccum genus. So further selecting these effective endophytes as a biocontrol, promising essential potential for management of mango anthracnose. Thank you.
Thank you. I have one doubt.
put
Uh,
fruits also.
Fruits.
Yes.
Yeah. Okay.
Okay, thank you.
Thank you, sir.
Now I will call upon Vidya Kamal Rathod.
Morning. Good morning, everyone. I, Vidya Rathod, Senior MSc student from the Department of Plant Pathology from the University of Horticulture Sciences, Bagalkot. I am here to present on the topic of biorational approaches for the management of wilt complex in Gava. Coming to the introduction, the Gava is a tropical fruit scientifically called as a CDM gajava, belonging to the family Myrtaceae. The Gava wilt is more complex due to the multiple pathogens such as Fusarium oxysporum pharma species, Melodogyne, and Rhizoctonia solni. Due to the complex nature, it will be managed to the difficult. With objective, the isolation and characterization of a pathogen associated with the wilt diseases of GAVA and isolation and characterization of a bioagents from GAVA and their evaluation against the GAVA wilt pathogen. As we can see that symptoms of GAVA wilt disease, first initially yellowing of leaves, drying up later, the drying of complete plant, and below ground symptoms are nematode infestation that will lead to the gall formation on the root surfaces. Coming to the materials and method with regards to the pathogen isolation, firstly we collected the infected root samples. Later we isolated the fungal pathogen through the standard isolation procedure and also isolated the plant parasitic nematode through the Biermann's funnel technique. And later we carried out the purification, characterization and identification based on the molecular and morphological characteristics. Coming to the pathogen identification and confirmation of a morphological and molecular characterization based on the culture of Fusarium oxysporum pharma species CD, which consists of three kinds of spores such as chlamydospores, microconidia, and macroconidia. Coming to the molecular characterization of pathogen, the PCR amplification was done through the ITS region, and we also constructed the phylogenetic tree of Fusarium species based on the ITS sequences. Coming to the nematode identification, we can observe here the nematode juvenile and plant parasitica nematode consisting of a silate, egg of nematode, and also we done the perennial pattern of nematode, which indicates that the Melodogyne species. Coming to the materials method with regards to the endophyte, firstly we collected the healthy GAVA plant tissues such as leaf, petiole, flower, and fruit, and later we isolated the bacterial endophytes, and we can do the purification and characterization through the morphological and microscopic observation, and later we done the in vitro screening through the dual culture technique, and later we evaluated the pot culture through the pathogen identification. Coming to the results, the passport data for bacterial endophytes from GAVA, here we isolated the 32 bacterial endophytes from the different varieties such as Taiwan pink, BNR, L49, Allahabad Saped, and many others, which offer different plant parts such as leaf, petiole, flower, and fruit stalk. The in vitro evaluation of a bacterial endophytes against the Fusarium oxysporum pharma species CD, here among bacterial 32 endophyte isolates, which UHSB G22 showed the highest inhibition of 72% and lowest mycelial growth of 25 mm showed compared to the control. Coming to the morphological and cultural characteristics of a bacterial endophytes, that is UHSB G22, here the biochemical test characterized through the high assorted biochemical test kit with consisting of a 12 tests, such as the positive reaction was showed in citrate utilization, lysine utilization, ornithine utilization, and urease and lactose. And later we have done the gram reaction, that is UHSB G22 showed the positive reaction. Later, we have done the same analysis of a isolate, that is UHSB G22. Here we can observe that the bacterial population was clearly observed in the rod shape. Coming to the population dynamics of a TAC bioformulation, that is UHSB G22. Here in this graph, we can observe that the bacterial population was gradually declined over a period of time, and later the bacterial population was maintained at 75 days. This result indicates that the bacterial population was at 75 days was survival. Coming to the pot studies here, the total treatments were 8 treatment was control, and T7 treatment, that is soil trench of a carbon diazine and mancozeb with a UHSB G22 showed the highest disease reduction and lowest disease index, followed by T5 treatment, that is carbon diazine with a UHSB G22 showed the highest reduction, that is 85%, lowest disease index, that is 13% compared to the control. With this, I conclude that the Gawa wilt disease is a most complex disease that involving the multiple pathogens which will be difficult to manage. Therefore, the endophytic bacteria, that is UHSB G22 showed the highest inhibition against the pathogen, that is Fusarium, and the integrating effective bioagents with a suitable management practices can provide a sustainable approach for the management of Gawa wilt complex. Thanks.
Thank you for nice presentation. It is one of the most important that is a wilt complex. It is mainly associated with the Fusarium, rhizoctone. So you have worked very nice, and I have one doubt. Efficient endophyte. So I have to mention which endophyte.
Sir, the research work is still going on, sir.
Okay. Thank you.
Thank you, sir.
Now the topic is open for discussion. If any clarifications.
So isolate that you have taken actually, will it fight back the melodogain also?
Sir?
Will it counter the melodogain also?
Sir.
Did you try in the field? I mean, like in the
So melodogyny was
lab
done,
conditions, have you tried?
done, sir. Yes, sir, I will try it, sir.
No, have you done it? You will try it.
Sir, we have done, sir.
We have.
Yeah, Shruti, how did you confirm that that your strain UHSB G22 is a unique
Based on the in vitro evaluation, sir,
in vitro elevation? So have you sent it
against
to
pathogen.
no? Have you sent it to any the laboratories for confirmation?
Sir, we have sent it, sir.
So
The result will confirm, sir.
okay. So, but based on your in vitro evaluation, it is you are saying that it's unique.
Yes.
Yeah. Okay.
Okay, thank you.
Yes.
Now I will call upon Krapa KS is going to present on unraveling the Ridgegard virome genome and development of rapid detection assays.
Very good morning to all. World of mesobiotic. Where unrolling the Ridgegard virome through NGS and development of rapid detection assays Reeds gourd is one of the important vegetable crops grown in India. It is affected. It is have its own medicinal and industrial and nutritional value. It is affected by many diseases. Of the different diseases, viral diseases playing important role for reducing its yield and quality. Where you can see many viruses like watermelon, butternut necrosis, cucumber mosaic virus, cucurbit, stunting virus, Jacini yellow mosaic virus are the major virus associated Reeds gourd in the world and also in India. But there is no comprehensive data in which we can detect a virus in a single reaction. That's why we have, in this backdrop, we have taken work on it the following objectives: perform virome profiling in the Reeds gourd, identification of all viruses in the Reeds gourd, and development of a nucleic acid based tools for rapid detection of virus associated Reeds gourd and their validation. Initially, we have surveyed this and for collected samples for the virus infection and also for the presence of virus. This is the workflow where we have, for the virome profiling, we have to collect sample, RNA extraction should be done. Then we have carried out the quality of the RNA was checked, the pooling of the RNA from the different samples and sent for sequencing in the library preparation and sequence analysis. After sequence analysis, we have used these standard bioinformatics tools for identification of virus and also identify recombination in the virus. After identification of virus in the virome data, we have standardized two tools, that the RT-PCR and RT-LAMP assays for detection of virus associated with Reeds gourd. These are the different software we use for the bioinformatics analysis of the data generated from the Reeds gourd virome, where we can see quality and also assembly and also search for the virus and phylogenetic analysis to determine the phylogenetic analysis and recombination of the virus within the virus. Then we also two assays are developed. This is the RT-PCR, where we have designed the primer for the pi virus identified in the study by using coat protein, also the nuclear settled protein in case of watermelon bud necrosis. Then quality of the primers are checked with the standard protocols. Like that, we have designed a primer for all the pi virus. These primers are used for the RT-PCR assay. Then like that, RT-LAMP, it is a unique, RT-LAMP assay is a unique where we can avoid the usage of PCR and also we can avoid the running gel also. We also designed a primers for the all fibers identified the virome data. This is the representation of the one representative of the virus primers designed for the RT-LAMP assay. These are the different viral infection symptoms associated with Reeds gourd in where surveyed area. We able to see the incidence up to 70% in the different location in southern Karnataka, indicating this what are the hybrids grown in southern Karnataka are susceptible and have varied symptom associated with Reeds gourd. Then after, this is the gel photo showing the RNA extraction of the 40 samples surveyed and collected. The best quality, all the quality was checked. The equal molar concentration of RNA was pooled. The pooled sample was subjected for the analysis in order to identify the quality of the RNA. We able to get a very good RNA. That RNA was subjected for NGS analysis, where we able to get very good data for the after sequencing. The data was streamed and used using a different software. We able to identify the 5 viruses associated with Reeds gourd first time in the country, where we were able to recover the 95% of the genome in the virome data. This able to identify these 5 viruses associated with the Reeds gourd. One is the DNA virus, 4 are RNA viruses. We established a phylogenetic relationship for tomato leaf curl virus, and also these tomato leaf curl virus DNA A has associated the recombinant, and DNA B also we analyzed such that this is the recombinant virus. Initially, we have established this association with the yellow mosaic disease in Reeds gourd. Then it is another first report in the India, cucurbit aphid-borne yellow mosaic virus is a pulmonary virus associated with most of the cucurbids. We able to establish system with the Reeds gourd also. Here no recombination of virus. Then this watermelon bed necrosis also associated with the Reed's guard with all segments of watermelon bed necrosis was established also with the recombination. This is a medium segment of the watermelon bed necrosis and larger segment of the watermelon bed necrosis virus. We are able to check the recombinant in this virus also. This is the papaya ring spot virus associated with the Reed's guard. The first time we have reported from the virus of the Reed's guard. There is a recombination in this virus. This is the tomato mosaic virus also associated with the Reed's guard. We unable to get the recombination in this virus. Based on this, we optimize the RT-PCR the full sample. This RT assay was used for validating the different samples were collected in the field samples. We able to detect more number of samples the tomato leaf curl virus and cucurbit aphids-borne virus also. These all viruses in the different capacity we able to detect with the RT-PCR. Later we also optimize the RT lamp assay where we can avoid the usage of a what is a PCR, and a field level application can be used by naked eye using intercollecting dye. Next, these are the RT lamp analyzed samples. We able to detect more number of samples as compared to the RT PCR assay. For that, all 5 viruses was used for detected through using the RT lamp assays using specific primers to the particular viruses. This comparative analysis with the RT-PCR, RT-lamp indicating the more number of samples were detected in case of RT-lamp, indicating this is a more sensitive in detecting the any pathogen, including virus. And these RT-lamp and RT-PCR analysis indicated that there is a multiple infection with respect to Reed's guard. This is useful. These assays are useful for detecting the different viruses in Reed's guard. Discussion, there is an incidence of a virus was observed, and it will have a population, depending upon the vector population, hormonal condition, cultivar, and stage of the crop, and cropping pattern, and practices followed. Then NGS revealed the complex of viruses. This NGS will helpful for the discovery of new viruses also. RT-lab, RT-PCR as used for can be detection of multi-infections in its guard. RT-lamp, already I mentioned, is a more enhanced potential to rapid diagnosis of the viruses. Finally conclude that rowing survey indicates the presence of viral disease up to 70% in the Reed's guard. The Reed's guard has showed various symptoms associated with the virus infection. NGS data indicated the presence of 5 viruses, and there is a recombination of 5 viruses associated with Reed's guard. RT-PCR and RT-lamp assays are developed for detection of the 5 viruses. RT-lamp more sensitive, detecting more number of samples as compared to the RT-PCR. These both assays has revealed the association of multiple infection in the viral infection in the Reed's guard. Then finally, I recommend that refining these diagnostic tools for higher sensitivity and field applicability, proving pathogenicity for the identified viruses, integrating these virome data into the breeding program for identifying virus resistance varieties, exploring these plant emerging viruses and epidemiological impact. These are the recommendations. Finally, I should acknowledge Government of Karnataka, Government of India for funding archive scheme for establishing a highly sophisticated diagnostic laboratory in our university. I thankful to the University of Artificial Sciences, Bagalkot, giving an opportunity to work and establish my, I'll say, pathologist. Thank you, Nandal.
Nice presentations and valuable informations with regard to the most important serious diseases in our Reed's guard, that is Reed's guard yellow mosaic. The incidence of the disease, it may range from 70 to 100% sometimes. So you have worked very well with regard to the important, especially in the cucurbits, and also you have developed your own techniques for the detection of the viruses. That is a lamp that is more sensitive and valuable confirmation of the viruses. So I'm very happy that for your work is your very good progress. And also I have one doubt with regard to the tolerant cultivars against the Reed's yellow mosaic disease.
Sir, Reed's actually in worldwide, no, we have evaluated more than 150 germ plasmid received from LPPGR, no resistance varieties. See, now we are looking for the collaboration with biotechnology, looking for susceptible genes against chromatoliphical needle virus. Because no resistance means we have to use other genomes like the genome editing. Susceptible means we are one of our PhD student has cleared the transcriptome where the susceptible genes are play important role. As Dr. Raghavendra Gunna is working on susceptible genes pomegranate, same approach we are exploiting in Ritzgard also, sir. But there some tissue culture techniques are required to establish it to transformation also. Sir.
Thank you. Now the topic is open for discussion. If any queries? Thank you, Shekhar.
Thank you. Sir.
Now I request Yoganand T is going to present on blue biotechnology for crop protection, seaweed, endophytes, and metabolites based solutions against thrips.
Good morning, one and all. Nowadays, farmers are rapidly using seaweed extract because of their biostimulant property, and some of the research studies shows that these seaweed extract are also having insecticidal properties. Our research journey starts with a simple and intriguing question that what if the biostimulant and insecticidal property of this seaweed extract, not only because of the biomass of this seaweed, but also due to the presence of endophytes in it. So we designed the research to the ongoing problem that is Thrips parrispinus. Back. Back. Because India is the major producer, exporter, and consumer of chili, due to the invasion of this chili Southeast Asian thrips, that is Thrips parrispinus, the chili yield was reduced by 50 to 80%. To overcome this, farmers are majorly dumping chemical insecticides, which not only affect the environment but also increased the cost of cultivation. To overcome this, we plan a research work which bridges the marine biodiversity with sustainable pest management. It's my pleasure to share my research journey with you. We conducted a roving survey around east and west coast of southern India. We collected marine algae from Rameshwaram, Kanyakumari, Kochi, Goa, St. Mary's Island, Karwar, etc. We collected these marine algae in sterile containers containing the local seawater and brought to the laboratory. After bringing to the laboratory, we isolated the endophytic fungi from those marine algae and conducted bioassay and larva of Spodoptera litera. We isolated so many fungus. Among that, 9 were promising isolates of endophytic fungi, among which Aspergillus and Penicillium are the given more results. Simultaneously, these marine algae were sent to a marine algal research station for their identification. Among the marine algae, we collected 4 are red algae, 4 brown algae, and 3 green algae species. Further, these algae were shade dried at room temperature, powdered, and the metabolites were extracted by using a Soxhlet apparatus. Frequently, fungal samples were prepared and isolated the metabolites. For metabolic profiling, we use GCMS and LCMS. This is the GCMS profiling of marine algae, which shows the plenty of bioactive compounds, which are having antimicrobial, insecticidal, and biostimulant property. Similarly, LCMS of marine algae shows that these compounds belong to different groups, that is fatty acids, terpenoids, phenolic compounds, and other secondary metabolites. Similarly, we conducted GCMS and LCMS of fungi. With the availability of chemical compounds in India and cost and the benefits, we screened some of the compounds and tested on Spodoptera littoral larva, among which D1, D2, S1 and S2 give the mortality of more than 90% on Spodoptera littoral larva. With the compatibility, persistency, and phytotoxicity data, we combined D1 and D2 and formulated as UHSB DD. Similarly, we combined and formulated as UHSB SS and conducted a laboratory bioassay on Thrips parvis pinus through flower dip bioassay and residue contact method. Simultaneously, we conducted assessment on bioassay on Bacan bavicornis, that is a beneficial parasitoid. Coming to the result, UHSB DD shows the significant mortality of more than 60% after 24 hours after treatment in flower dip bioassay method. And similarly, in residue contact method, around 58% mortality is observed at 2000 ppm. Similarly, another product, UHSB SS, also showed the mortality of more than 50% at 2000 ppm after 24 hours after treatment. Next. Coming to the survival rate of Bracken bavicornis, there is no much significant difference between the treatments and the control condition. And this Bracken bavicornis survived more than 60% 3 days after treatment, even at 2000 ppm concentration. Further, we are now doing the field studies of these compounds in the field level condition. And also we are doing gene regulation studies in chili plant. I would like to conclude my topic by saying that marine algal derived metabolites demonstrated the strong bioefficacy against Spodoptera littera and cause a significant larval mortality, growth inhibition, and critical deformities. This promising activity of UHSB DD and UHSB SS against Thrips parvis pinus highlight the potential of CV-derived metabolites to transform marine algae into green production of chili. And I would sincerely thanks Anusandhan National Research Foundation and PM Fellowship Scheme, and equally to the Pinnacle Biosciences for the collaboration for my research study. Thank you.
Thank you, Yoganand. What are all the components involved in the metabolites?
Metabolites, I noticed so many like fatty acids, terpenoids, secondary metabolites, some phenolic compounds, etc. Alkaloids.
Okay. So any questions from delegates? Audience. Okay, if there are no questions, so what is the basis of mixing DD, two compounds in DD and two compounds in SS?
Sir, based on the phytotoxicity and their compatibility studies, we mixed that one.
But still then the mortality, what we are seeing is around 50%.
No, sir, that is mortality at 24 hours after treatment only, sir. After that, the mortality increases, sir.
So that you are not shown.
No, sir. We evaluated because thrips in control condition also may die after 24 hours in the laboratory condition, sir. That's why we restricted to 24 hours. That's
okay. So any questions? Okay, thank you. Thank you, Dr.
Yes, sir. We multiplied that one, sir. We actually seen the metabolites which are present in that aspergillus and penicillium, sir. Those metabolites are not present in the what are available in the land aspergillus and land penicillium. Means the endophytes which are present in the land plants and the marine endophytes are completely different, sir. Yes, sir. We didn't apply the fungus itself, sir. We isolated only the metabolites, and those metabolites are testing on the insects.
Yeah, thank you, Dr.
Thank you.
So now we have one the online presentation, Dr. Sangeeta CG. That is on morphological characterization and in vitro.
Aspergillus and penicillium, sir. Those metabolites are not present in the water available in the land.
So that is Sangeeta, is she available?
Yes, sir. Yes, sir.
So the topic is morphological characterization and in vitro evaluation of indigenous
Didn't apply the fungus itself.
against stagnosporosis, cucurbitacearum. Dr.
Thank you.
So now we have one
Yes, sir.
Sangeeta.
I am, yes, sir.
Dr.
Yes, sir.
Sangeeta CG.
I
That
am
is
unable to share my, this one.
Aspergillus and penicillium, sir. Those metabolites are not present in the
stagnosporosis. Dr.
Thank you.
So now we have one.
Dr. Manjunath. So you are coming to end of the session, ecological pest and disease management in horticulture. I sincerely thanks to the organizer for giving an opportunity to chair the session. Now I might hand over to our co-chair, Dr. Venkatreshulu.
So thank. In the beginning, we had very good leads, the talks by Dr. Prabharaj from US Raichur, and also Dr. Raju Tegeli for presenting a very nice impact of the NICRA project. So I thank the lead speakers and also the presenters. Very good presentations we had. So I thank the organizers for the opportunity. Thank you, sir. I thank the rapporteurs. Thank you very much.
Thank you, sir. Now I request Honorable Vice Chancellor for issuing a memento and certificates to the chairman and co-chairman of the session.
So you are coming to end of the session, ecological pest and disease management in horticulture. I sincerely thanks to the organizer for giving an opportunity to chair the session. Now I might hand over to our co-chair, Dr. Venkateshwaru.
So in the beginning,
Now I request the Honorable Vice to certificate for the rapporteurs as well as the moderator. So now we'll break for 5 minutes for tea and then again reassemble after 5 minutes. For the dignitaries, tea is arranged in the lounge here adjacent to auditoriums. Just sorry,
Check. Hello, hello. Check. Hello, hello, hello. Leave happy. Hello, hello.